I'm Tori.
I'm Brendan.
I'm William.
I'm Allie.
I'm Zach.
And I'm Greesh. And from our experiment, we have found that in vivo addition of 400 nanomolar DES1 alpha factor decreases mating response in Saccharomyces cerevisiae strain LM233A in absence and presence of 400 nanomolar wild-type alpha factor. Signal transduction is the transmission of molecular cells from the cell's exterior to the interior. It is especially important in biological processes such as cell development, cell reaction to stimuli, and intracellular communication. To understand signal transduction, we will be looking at the mating pathway in haploid Saccharomyces cerevisiae. The ligand for this pheromone pathway is alpha factor. When the alpha factor binds to the receptor STE2 protein, the ligand receptor complex signals the dissociation of the G protein complex into alpha and beta gamma subunits. Activation of the beta gamma subunit results in morphogenesis of cells indicated by the shmoo shape and subsequently initiates the MAPK cascade. This results in cell cycle arrest. This process also stops budding and increases the number of cells displaying unbudded phenotypes and transcription of mating gene fuse1. Inactive STE2 protein is found in dimerized form. The two monomers here highlighted in green and pink have disulfide bridges that link their TM5 and TM6 regions together. The left image shows the conformation of the TM5 and TM6 regions of one STE2 protein receptor when it is in the dimerized state. When alpha factor binds, the bridges are disrupted along with the dimerization as the TM5 and TM6 regions undergo conformational changes. This activates the mating pathway as shown on the image in the right. The alpha factor consists of three functional domains. At the N-terminus, first 4 amino acids are associated with the initiation of signal transduction from STE2 receptor. 7 through 10 amino acids form a loop domain that is involved in maintaining proper orientation of the ligand that supports proper binding and signal transduction. The last 4 amino acids at the C-terminus of alpha factor are involved in securing ligand to the receptor. Since we know that the structure of alpha factor is important for inducing conformational changes in the receptors, we were curious to see what the effect of modifications in this alpha factor structure might lead to. We used a mutant analog of alpha factor which has deletion of the first tryptophan residue in the N-terminus. Our study question is, what effect does the DES1 alpha factor, a mutant analog of alpha mating factor, have on S. cerevisiae A cells in the presence and absence of wild-type alpha mating factor? And now, Zach will dive into interactions between alpha factor and STE2P receptor.
Thank you, Greesh. This image represents the interaction of the N-terminus of alpha factor with the STE2 receptor. The STE2 receptor itself is shown as coiled ribbons, with the yellow ribbon representing the 5th transmembrane alpha helix and the orange ribbon representing the 6th transmembrane alpha helix. Alpha factor itself is represented with a purple ribbon, with residues of importance shown by ball and stick modeling. The first tryptophan of alpha factor interacts primarily with tyrosine 266 and lysine 269 of the STE2 receptor. The first tryptophan of alpha factor, in conjunction with the third tryptophan of alpha factor, interacts with tyrosine 266 to form a pi-electron stacking motif. This motif provides stability of the binding of the N-terminus of alpha factor with the STE2 receptor. Without the first tryptophan, this pi-electron stacking interaction will be severely reduced. Therefore, we assume that the overall geometry of the N-terminus of alpha factor relative to the STE2 receptor will be changed, and that this change will result in overall less signal transduction. Additionally, the first tryptophan of alpha factor interacts with lysine 269 of the STE2 homodimer, as shown in the image in the far left. Normally, this lysine interacts with counterions that prevent it from interacting with other portions of the STE2 homodimer. However, the binding of alpha factor leads the first tryptophan of alpha factor to disrupt this interaction, freeing lysine to reduce disulfide bridges between the homodimer. This results in a pushing apart of the two monomers to form the active STE2 receptor. Without the first tryptophan of alpha factor, this interaction will be severely curtailed, and that this will also contribute to reduced signaling transduction. Therefore, we hypothesize that the binding of DES1 alpha factor lacking the first tryptophan will result in less shmooing, less cell cycle arrest, and less fuse1 gene transcription than the binding of wild-type alpha factor. Additionally, we hypothesize that the simultaneous addition of wild-type alpha factor with DES1 alpha factor will also result in a reduced mating response. This is assuming that the amount of alpha factor added with single alpha factor treatments is sufficient to saturate the receptor. And now Allie will describe our general methods.
To set up our experiment, we created an overnight culture 18 hours before we began our assay.
Then we transferred our overnight broth to create 5 sets of dilution replicates at a 1:9 dilution ratio of the overnight culture to the SD-0 broth. Uracil lacking media is utilized to serve as a selection agent against yeast not containing the PBH315 plasmid. This plasmid also contains LAC-Z associated with FUSE1 promoter to allow the measuring of beta-galactosidase as a proxy for FUSE1 gene expression. 4 hours prior to the assays, we grew each set of dilution replicates for 2 hours, staggering our addition of alpha factor by 45 minutes to allow for sufficient time to perform the cell shape and beta-galactosidase assays while maintaining a 2-hour exposure to alpha factor. Each set of dilution replicates was divided into 4 treatment conditions shown here. Minus represents the negative control with no alpha factor. Plus represents the positive control with wild-type alpha factor. Minus plus represents DES1 alpha factor, and plus represents wild-type and DES1 alpha factor. We collected 2 sets of data but excluded the first set for 2 reasons. The first was that we didn't stagger our conditions appropriately, so replicates were not all exposed to alpha factor for 2 hours, making the dilution sets incomparable. Second, the first set of data had a high standard error, likely due to experimenter inexperience. Therefore, we excluded the first set of data to minimize human error and to be able to better focus on our biological system. We performed 3 assays, a cell shape assay, a beta-galactosidase reporter assay, and a Western blot assay. We'll start by describing the methods and results of the cell shape assay. For the cell shape assay, observers were blinded to the identity of treatment conditions during observations of unbudded, budded, and schmooing phenotypes. We analyzed these results with the chi-square test of independence. Illustrated here is a graph of our cell shape assay results. Our y-axis represents the percentage of our yeast phenotype, and our x-axis represents the treatment condition. The darkest shade of purple represents budded cells, the intermediate purple represents unbudded cells, and the lightest purple represents schmooing cells. As a reminder, the minus has no alpha factor, the plus has wild-type alpha factor, the minus plus has DES1 alpha factor, and the plus has both. The 3 treatment conditions with alpha factor had a significantly larger unbudded to budded cell shape ratio and a larger percentage of schmooing in comparison to the negative control. The positive control had a significantly larger unbudded to budded cell shape ratio and a larger percentage of schmooing cells than the negative control. Treatment with DES1 alpha factor had a significantly smaller unbudded to budded cell shape ratio and a smaller percentage of schmooing cells than the positive control. However, treatment with both alpha factors did not yield any significance, but did show a trend of a larger unbudded to budded cell shape ratio and a smaller percentage of schmooing cells compared to the positive control. Now William will elaborate on the beta-galactosidase assay methods and results.
Thanks, Allie. Prior to each assay, we measured the cell densities of each of our 4 replicates by recording their absorbances at 600 nanometers. By doing so, we ensured that their yeast cells were in the log phase of growth, which is when they are most responsive to the addition of mating factor. Next, we exposed the yeast cells to a solution of Z-buffer, chloroform, and 0.1% SDS, which facilitated the beta-gal reaction with ONPG in the next step by increasing the permeability of the yeast. The colorless ONPG was then added as a substrate for beta-gal because its cleavage by beta-galactosidase forms a yellow O-nitrophenol product during the reaction. Once the first significant yellow color change was detected as compared to a non-transformed yeast control, or after 5 minutes had elapsed, the reaction was stopped with sodium bicarbonate. Finally, absorbances to detect the amount of O-nitrophenol product produced were measured at 420 nanometers. The absorbances and reaction time were converted to a comprehensive milliliter measurement and analyzed with one-way ANOVA and unpaired t-tests. This graph displays the results for our beta-galactosidase reporter assay, with error bars representing plus and minus 1 standard error of the mean. Again, the x-axis represents treatment group. From left to right, we have our negative control, our treatment with just wild-type alpha factor, with just the mutant DES1 alpha factor, and finally with both wild-type and mutant DES1 alpha factor. The y-axis represents Miller units. Overall, we found that beta-galactosidase activity depends on treatment group, as evidenced by a comprehensive one-way ANOVA. Next, we investigated the effects of the 2 mating factors on beta-galactosidase activity. First, we ran a one-way ANOVA between our 3 groups with mating factor and found that the mean beta-galactosidase activity was significantly different amongst them. Based on this finding, we took a more specific look at whether the mutant DES1 alpha factor showed reduced signaling as compared to the wild-type alpha factor, which was our positive control. When we compared the wild-type and DES1 only groups through an unpaired t-test, we found that the DES1 mutant factor produced significantly lower beta-galactosidase activity. Finally, we were ready to evaluate DES1 alpha factor's ability to act as a competitive inhibitor to wild-type alpha factor. To this end, we compared the DES1-only group with the combined DES1 and wild-type alpha factor treatment group. While an unpaired t-test showed that these groups were not significantly different, this result is affected by the high standard error in our wild-type data. Our overall trends show that the beta-galactosidase activity was highest in the wild-type alpha factor group. This was followed by the combined wild-type and DES1 alpha factor group, and then the DES1-only group. This is important and most likely biologically significant because these results were replicated in our Western blot assay, which Brendan will discuss next.
Thank you, William. So with regards to the Western blot assay, we ran SDS-Page using a 10% polyacrylamide gel following digestion with a 2x lamelli buffer. The gel's protein content was then transferred to a nitrocellulose membrane using a Biorad transblot turbo system. Beta-galactosidase was visualized using a Promega mouse anti-beta-galactosidase antibody for the primary antibody, followed by a Promega anti-mouse antibody conjugated with alkaline phosphatase for the secondary antibody. Here we have the results we obtained for the Western blot assay. On the right is the nitrocellulose membrane, with visualized beta-galactosidase shown within each rectangle of the overlay. Note that we did not observe any band in the negative control lane, as expected. On the left, we have a graph representative of the intensity of each band shown in the nitrocellulose membrane. The x-axis depicts treatment groups, while the y-axis depicts beta-galactosidase to total protein content per lane. Two essential manipulations on the data were performed in order to best present it. The first data manipulation was to create a ratio comparing beta-galactosidase to total protein content per lane. Total protein content was quantized with Biocore image software via UV irradiation images. The second data manipulation was to create a ratio which compares all values against the positive control for ear-to-ear comparisons. Thus, the positive control is displayed as 1, while the minus plus group is displayed as 0.23, and the plus plus group is displayed as 0.7. As there was only one replicate for each condition, we did not run statistical analyses on this data. However, trends seen in the Western blot data are very apparent and support the results from the previous two assays. Now Tori is going to continue on with our discussion.
Thank you, Brendan. Our hypothesis that deletion of the first tryptophan in the DES1 alpha factor would lead to reduced mating responses compared to the wild-type alpha factor was supported by the results in all of the assays. The assumption that the interaction of the first tryptophan with tyrosine 266 is important for activation of STE2P is supported. Although the third tryptophan also interacts with tyrosine 266, the reduced signal transduction when the first tryptophan is deleted suggests that the third tryptophan alone is not able to as effectively activate tyrosine 266 as both tryptophans are together, supporting the model where the indole side chains of the tryptophans form a stable conjugated electron system with tyrosine 266's aromatic ring. Without the first tryptophan, the stability of this interaction is disrupted, decreasing signal transduction by changing the orientation of the alpha factor relative to the receptor. These results also support the idea that the first tryptophan's interaction with lysine 269 plays a significant role in signal transduction. It is well established that lysine 269 is important for effective signal transduction, but these results also support that without the first tryptophan, lysine 269 is less able to disengage with counterons, and thus less able to disrupt the disulfide bonds in the STE2P dimer, preventing its activation. Our hypothesis that the mating response of the alpha factor with DES1 alpha factor would be reduced compared to the alpha factor alone was supported by the trends in the assays. Although these trends didn't reach statistical significance, that all three of the assays had the same trends increases our confidence and makes us believe that these trends are biologically significant. The mutant alpha factor's ability to competitively inhibit the effect of the wild-type alpha factor indicates that the mutant ligand was still able to bind to and block the receptors, but was not able to induce signal transduction as effectively as the wild-type alpha factor. This indicates that the first tryptophan is not important for the alpha factor's binding affinity, supporting the concept of the signaling domain being primarily involved with signal transduction. To further explore our study question, a higher replicate number and a larger sample size may make trends in the data more detectable. Currently, our confidence is reduced because of our small sample sizes and results that did not reach statistical significance. Having a higher replicate number would increase statistical power and our confidence that our results are representative and our conclusions are appropriate. Further, mutations in the residues lysine 269 and tyrosine 266, in conjunction with our treatment conditions, could contribute to our understanding of how important the first tryptophan's interaction with each of the residues is for signal transduction. In conclusion, our study found that the deletion of the first tryptophan reduces signaling activity. This supports the importance of the first tryptophan's interactions with tyrosine 266 and lysine 269. Our study also found that the deletion of the first tryptophan does not reduce binding affinity, which supports the idea that the signaling domain is primarily involved with signal transduction. Thank you.